Hey guys, Dr. Mobeen. So we are talking about the genetics of immunology. In our previous lecture, we just finished talking about the genetic rearrangement that helps to create diversity in the antigen binding site. This is the last topic of that topic, and that is the somatic hypermutation. Again, as I have been saying, please understand and study the genetics of immunoglobulins or the diversity of the immunoglobulin in following ways. This is the genetics before the immunoglobulin structure starts forming. Here the DNA itself is shuffling itself and rearranging itself. Then the DNA is finalized, messenger RNA is formed, and it is making proteins. These proteins make light and heavy chains, and then in various combinations of light and heavy chains, we end up with more diversity. And finally now, somatic hypermutation is that light and heavy chains have become combined. So light chain, heavy chain have become combined, and the cell is actually functioning and it is giving immunoglobulins off. Even then it still keeps learning. Just like when we are born, we have a set of knowledge, and then as we keep growing, we keep learning. We don't just stay there. As you are medical students or doctors, you're continuously learning. So the same thing is true for the B cell. B cell continues to learn. The only thing that is funny, I always like it, just like natural killer cell, I think it's a pervert who touches every other cell. The B cell, which I find this somatic hypermutation is a very, it's a mechanism which has love involved in it. So this is actually a love story of a B cell and a follicular dendritic cell, and that love story actually helps B cell become more sharp in its killing mechanisms. So let's see the love story. Let's see how this works. So let's say we have a B cell or a plasma cell, and this is actually an active cell. Has DNA. In this DNA there is a V region, a D region, a J chain region for the heavy chain. Then one thing I forgot to mention in the previous one is that for the light chain variable region we only have V and J. I think I said it. So these genes are there. Then of course we know that, let's say in our case it is constant mu region is attached here. So the constant mu is the heavy chain. So this DNA is going to make IgM. Depending upon what kind of a poly A tail is attached here, hydrophilic or hydrophobic, this will be the IgM created will either be stuck on the surface or it will be released. So let's say in our case we release this IgM. Why? Because we want to do something with this. So this IgM is released, and actually I wanted to say that let's say it stays with the cell membrane here, IgM. So you know that the IgM that is sticking with the cell surface membrane, B cell surface membrane, acts as a receptor. We saw that it acts as a receptor for antigen presentation function or APC function. Now we'll see another function of IgM that is sticking on the cell surface. This is the love story. Now this cell is going to be, so this guy, this cell or girl doesn't know that dendritic cell loves it. So let's make a dendritic cell. So here is a dendritic cell. So of course you know it's a dendritic cell, it has dendrites. So this dendritic cell is totally in love, and it's always looking for the B cell, right? So this B cell has gone in the lymph node. It is an active B cell. It is releasing IgM. It has IgM on its surface. So it is actually, it has done some antigen recognition, and it is functional. It is fine. It has recognized antigen. It has made an immunoglobulin that can bind to an antigen. But this guy is funny. He always keeps presenting flowers to this one. So the flowers that the follicular, now remember this is follicular dendritic cell, not dendritic cell. What is the difference? The other dendritic cell which are present in the rest of the body, under the skin or the other tissues, they do not do this. This is the speciality of the follicular dendritic cells. So the dendritic cells that go in the lymph node, they fall in love. So lymph node is just so romantic. that the dendritic cell, once it is in there, it falls in love and it carries the flowers. Do you know what this flower is? This flower is a part of the pathogen. This is an antigen. So I'm just making up that they fall in love. They actually, what they do is, these dendritic cells, they eat up the pathogens, then they get the, they do the antigen presentation, and so by that process they have the antigen on their MHCs. These antigen are with what the B cell receptor will interact. So they just stand there. These guys just stand there with those flowers in their hand. They just stand there and say, try your receptor on this. So B cell comes near and it says, whoa, you got a flower for me. All right, I'm going to try my receptor. So this is the receptor, IgM or IgD. It is going to try to connect that to this antigen. Now when it connects it, remember this is already a good cell. It is already making IgMs. It knows how to connect with the antigen. It has formed a binding site that can connect. But now it is refining it. So it tries to connect with that. And based on the interaction here, the end result is that the conformational changes go all the way up to the nucleus, and there is something, the B cell's heart becomes really warmed that somebody gave it a flower. It really is like, it is really in love. Is that how they do it? They say, okay, there is hearts that appear. So what is happening? The heart of the B cell, the nucleus of the B cell, undergoes somatic hypermutation. Somatic hypermutation. And what does that mean? So now we understand the love story here. Now let's see what is going on in the heart of the B cell, the nucleus of the B cell, and what do we mean by the somatic hypermutation. So let me just tell it briefly and then we'll look at the mechanism. Brief thing is this, that these genes, they receive point mutations. Single nucleotide bases are mutated in these genes. So what? Does that make it better? Maybe not. But what happens is that every time when B cell interacts with this flower, with this antigen, with the follicular dendritic cell, there is one point mutation. That point mutation changes the structure of the next immunoglobulin slightly. It still binds to the same one. So if it was binding to the antigen like this before, maybe now it binds like this. But it is still binding to the same antigen. Binding pattern is changed, slightly changed. One nucleotide base change, it's not a big change. Now the binding may improve or binding may become bad. You know that the B cells work with this selection mechanism. What does that mean? That if there are, let's say this is the antigen, and if there are two B cells, this is one B cell and this is another B cell. So B cell A tries to connect with this and fails. It does not know how to connect. B cell B knows how to connect with this. Which B cell will become active? The B cell B. This is called the clonal selection. Clonal selection means that the B cell that would react with the antigen will become activated and selected to do the function. Now we are seeing another process of clonal selection. This B cell has already become selected, but now the copies of this B cell, one of those copies is going to be further selected. Why? Because every hypermutation would create a new B cell, because they'll keep proliferating, right? Because they're active. Every new B cell would have slightly changed immunoglobulin M or G or whatever. And that immunoglobulin would, when that would be attached to the antigen, if there is a good fit, that particular B cell would undergo differentiation and proliferation. And so out of all the B cell which can combine with the same antigen, the one which combines the best will be selected. Now let's see the mechanism. We understand the outcome. The outcome is that the B cell, the one which has gotten the best fit, will be selected to continue working. So this is sharpening the pencil here. Now let's see the mechanism. So the mechanism is that if I make a DNA here, so of course you know that these are base pairs in the DNA. So this is any one part of this DNA. So let's say this is this part. So this is the DNA, right? What happens is It's a very, very interesting thing. The guanine connects with the cytosine, right? So adenine and thymine and guanine and cytosine. There is an enzyme called AID, activation induced deaminase. What it does is it goes and steals an amine from the cytosine. I actually, just like these guys are in love, there are very interesting things happening inside, in the heart of the B cell. So the AID enzyme comes and steals an amine group. When it steals the amine group, the cytosine becomes uracil. So I know you're saying you cannot have uracil in the DNA. It is only okay to have that in the RNA. So yes, you're right. You should also be saying one more thing. Uracil does not connect with guanine, right? It does not. So what happened here? What we did here was we intentionally, this AID enzyme intentionally created a base pair mismatch. Why did it do that? Because it's a lazy guy. It doesn't want to do the rest of the things. It just created a mismatch. Now the repair enzymes would come and they would repair this place. You know how the repair would occur. What would happen is that once a mismatch has created, the repair enzymes would come and they would remove those base pairs, guanine and uracil, and they'll throw them away. Then the polymerases would come and they would put the new base pair here. And now is the chance to create mutation. When they would put the new base pair here, they may not put the guanine and thymine again. They might put adenine and cytosine here. They might put something else which is valid. And all of a sudden we've gotten one base pair change. This is a beautiful mechanism. So by this mechanism, now the next immunoglobulin which will be formed, that will be slightly different. This is called somatic hypermutation. Now remember this, that this somatic hypermutation actually becomes 10 raised to power 5 to 10 raised to power 9. 10 raised to power 5 is almost 100,000? 100,000 to billion times more frequent inside the cell once this cell activation and this mechanism is engaged. The end result is that the hypermutation would occur and slowly that wrench which is being formed to handle this pathogen antigen, that wrench would keep becoming better and better and better and better fit would occur, and then the antigen engagement will be better. Now this B cell which has gotten a good immunoglobulin connection will also then become memory B cell, and in future it will be used as well. So this is what is called the somatic hypermutation. Hope you understand that, and we will go to the class switching in our next lecture in a few minutes. Thank you.